Hello, my name is Megan Rath, and I'm the lead author on this project, The Relationship Between Plasma Tau Concentrations and Injury Status in Male Athletes Who Experienced an Acute Sport-Related Concussion. I am part of the Doctoral Integrative Exercise Physiology program at Temple University. We have many collaborators within our College of Public Health and the Lewis Katz School of Medicine. So sport-related concussion has been shown to be associated with future neurodegenerative diseases, including chronic traumatic encephalopathy, or CTE. And so we wanted to investigate the effect of acute sport-related concussion on plasma concentrations of brain-specific markers. And we used 4 markers in a multiplex assay so we could simultaneously measure them in each athlete sample. These markers included tau, which has been seen abnormally deposited in the brains of former athletes with CTE. Neurofilament light chain, this is NFL, it's an axonal marker. Glial fibrillary acidic protein, or GFAP, and ubiquitin carboxyl hydrolase L1. This is a neuronal marker. We abbreviate it UCHL1. Now, our participants were college-aged contact sport athletes. We best matched our athletes with sport-related concussion to healthy contact sport athlete controls. Now, for the controls, they had to be at least 1 year out from a prior concussion or head injury, and we collected blood samples for athletes with acute sport-related concussion within 48 hours of injury. We just have male athletes in this analysis, and so our sample size is quite small. It is n equals 12 for our group with concussion, and n equals 14 for our aged and history of concussion matched controls. We used the SIMOA, which is single molecular array platform through Quanterix in collaboration with the Alzheimer's Center at Temple. What we found was that plasma tau was significantly decreased within 48 hours of a sport-related concussion. Interestingly, none of the other markers had any significant difference between the athletes with sport-related concussion and our healthy controls. A potential reason for the decrease we see in tau is animal studies have found that in mice, plasma tau will be decreased in animals with dysfunctional or ablated lymphatic vasculature within the brain. So this could be a potential reason for the decrease that we see. However, we did not measure any markers of potential cerebral lymphatic vascular dysfunction. We also did not do any neuroimaging, so it's just an interesting concept that should be further explored. Also of note, we did have one athlete who experienced a very prolonged time until symptom resolution. So his 48-hour blood sample after injury was actually 3 times higher than the other athletes who sustained a sport-related concussion. So it is difficult to say with any certainty because it is only one athlete, but it is interesting if plasma tau levels that are extremely high could potentially be predictive of a prolonged or complicated recovery from sport-related concussion. So that is also something that should be explored. Now, since this was only conducted in male athletes, what we would really like to do is to increase the sample size and see if this phenomenon also occurs in female athletes, or if this is something that is predominantly specific to male athletes. we would also like to further study if athletes with this significant decrease in plasma tau following sport-related concussion also have some changes to their cerebral lymphatic vascular network, and if there is any correlation to that. I hope you enjoyed this presentation. Thank you for listening.